National 5 Biology Life on Earth video number 2, Distribution of Organisms. Biotic factors include competition for resources, disease, food availability, grazing and predation. Abiotic factors include light intensity, moisture levels, pH and temperature. If we look at predator-prey interactions over time, populations are not constant. Over time, the size of a population fluctuates. This green line shows the fluctuation in the numbers of prey, which is the hunted animal. And if we plot a line for a predator of this prey, it follows a similar pattern. However, the peaks and troughs lag behind the prey animal, like so. The population of each is actually related to the population of the other. As the numbers of prey rise, more food is available for the predators, so they too rise. Increasing predator numbers puts pressure on the prey animals, and their numbers begin to fall. Eventually, predator numbers fall enough to allow the prey animals to start to recover. And this pattern is repeated over and over, with the changes in predator numbers lagging behind those of the prey. We can measure abiotic factors using various probes and devices. When using a light meter, for example, the sensor should be placed at the same angle as the surface whose light intensity is being measured, and make sure not to stand in the way of the light meter to cast a shadow over it. If you're taking measurements over multiple days, make sure you take all the measurements as near to the same time of the day as possible, as the light intensity can change with the time of day or the cloud cover. If you're using a moisture meter, you should take the samples about 24 hours after any rain to allow any excess water to drain away, and make sure you insert the probe at the same depth every time you take a measurement. The probe should be wiped down to remove any dirt and moisture which may affect future results. When sampling organisms in the environment, or to estimate population sizes, one can use a quadrat. Basically a square frame of known area and divided into equally sized smaller squares. It can also be used to estimate the abundance or numbers of plants in an area. For example, estimating relative abundance, we place a quadrat on a lawn with different plant species in it. To estimate the relative abundance, we don't count the individual plants, we count the number of squares in which we find each species. So for example, if we're looking at the abundance of buttercups, we count the number of smaller squares that have buttercups in, and in this case, it's an abundance for the buttercups of 8 out of 25. If we count the other species in the quadrat in the same way, we find these results. When using quadrats for estimating population sizes or measuring abundance, it's important to throw the quadrat randomly and throw it as many times as possible. Transect surveys are used to determine how species change from one habitat to another. In this instance, the quadrats are placed at regular intervals along the line of the transect. This could be used to investigate the effects of light or moisture level changes on the abundance of plants in different habitats. Pitfall trap can be used to sample organisms living in a particular habitat. First, you dig a hole in the ground, then you insert some kind of small vessel, like a yogurt pot, for example. Make sure the top of the yogurt pot is level with the ground. Place a cover over the top to stop predators from coming along and eating whatever invertebrates you might find, and make sure it's camouflaged. Over time, small invertebrates will find themselves going into the pitfall trap and being caught there. One downside is, of course, if there are any large predators, like spiders, might fall into the pitfall trap, they might eat the smaller invertebrates. We use paired statement keys in order to identify organisms. If we take this bird, for example, if we work our way through the paired statement key, we can see that it has a non-red chest. So we go to question number 4. It has no blue in the feathers. So we then go to question number 6. And it does have a yellow patch on the chin, so we can identify those birds as bee eaters. A typical question you may get in the exam relating to paired statement keys is to be given a table of information and then being asked to complete the paired statement key. So why not have a look at the information here, have a go at answering 1 to 6, and then pausing the video and see if you get the answers correct. So the answers would be go to 4, then for question number 2, a black winged tip, and then go to 3. For question number 3, no spots on wings would be a wood white. For question number 4, a white wing tip would give you a red admiral. Humans can affect environments. For example, we can cause air and water pollution, and we can cause habitat destruction by deforestation, desertification, and by overfishing in the marine environment. The level of pollution that we cause in an environment can be found by studying the numbers and types of organisms found there. Species which, by their presence or absence, indicate environmental quality or the levels of pollution are called indicator species. If in this example we examine a river that's flowing downstream, and let's say there was a sewage works emptying its effluent at that point there, upstream of where the effluent was being emptied, we would find very clean water species, such as stonefly nymphs and mayfly nymphs. Immediately as the effluent enters the river, there's a complete change in the organisms found, and we find organisms that are used to very dirty water, such as sludge worm and the rat-tailed maggot. Further downstream, as the effluent is being broken down by bacteria, we find now dirty water species, such as waterlouse and bloodworm. Continuing on, we find cleaner water species, such as caddisfly larvae or freshwater shrimps. And then eventually, once all the organic material is broken down, we are back to finding very clean water species, such as the stonefly nymphs and the mayfly nymphs again. Burning fossil fuels, such as coal, oil, and gas, release sulfur dioxide into the air. And lichens are a group of plants that are sensitive to the presence of sulfur dioxide in the atmosphere. There's 3 main categories: crusty, leafy, and shrubby varieties that we find living on walls and off the bark of trees. If we look at the number of lichen species with increasing distance from the city centre, for example, the city centre would be the source of the sulfur dioxide pollution. Crusty varieties are found close to the city centre, so they are quite tolerant, or the most tolerant in this case, of sulfur dioxide in the atmosphere, followed by leafy varieties, and the least tolerant of sulfur dioxide are the shrubby varieties. So, with increasing distance from the city centre, we find a reduction in the concentration of sulfur dioxide in the atmosphere, coupled with an increase in the numbers of different